All right, so here is our official beginning. So again, I'm Colleen Hitchcock. I'm a member of the board of directors here for the Swampscott Conservancy. I want to thank everybody for joining us tonight for our webinar, What's That Smell with Don Pinkerton, and I'll introduce Don officially in just a second. Also, just an FYI for all of those who are joining us via Zoom, that we are in fact recording this Zoom meeting and we'll be hosting it either on our YouTube channel or via the town channel for later viewing. So you are all also being recorded. The Swampscott Conservancy, for those who hadn't heard of us before today's event, we are a small, relatively new nonprofit, just about 2 years old. And our mission is to support the conservation and open spaces of Swampscott. And the way that we do that is in a couple of ways. And you can see here we have a four-pronged approach to thinking about how to protect, enhance, and support our open spaces. And we do that through education programs, such as the one you are participating in tonight. We do that through citizen science efforts with a couple of approaches on that front, including documenting biodiversity with iNaturalist, doing some water quality monitoring. We also do that through habitat improvement and have a couple of ongoing projects that range from the pollinator garden down by Town Hall, which hopefully some of you have had a chance to enjoy this spring, that hosts native plants to support native biodiversity, removing invasives, and a restoration project currently underway at the Swampscott Middle School. And we do a lot of community building, getting out and together, which we're kind of do at a distance this summer, but from birding events to the farmers market and the like. If you want to become a more involved member of the Swampscott Conservancy, you're doing the right thing by joining us on this education webinar. You can become a member. We would greatly appreciate your support for free events such as this, participating in our education speaker series, doing some citizen science, or volunteering. And we currently have some volunteer efforts going on. If you are not already a member of our email list, shoot the Swampscott Conservancy an email and we'll add you to that email list, which will keep you up to date on the upcoming events and volunteer opportunities. So that's enough about the Conservancy. And so what we're here to do tonight is to hear from Don Pinkerton, who is a longtime Swampscott resident and has been involved in the community for a number of years in a variety of roles, as he discusses with us what is that smell, where we're going to discover local seaweed microecology. And for his day job, he is the biology and biotechnology teacher at Revere High School, and he's going to explain his work looking at the seaweed in Nahant Bay with some of his students over the past couple of years. And with that, I'm going to stop sharing and hand it over to Don.
Thanks, Colleen. It's nice to see a lot of familiar faces here. It's really great to see all of you. And let's see how this goes. I'm going to be showing some slides and sharing my presentation in just a second. I do want to give a disclaimer. It's a couple of things. Despite the title, what is that smell? I'm not personally responsible for that smell. So if you're smelling something, that is not me, but I just thought it was sort of a catchy title that Colleen attributed to this. Thank you. And also, I want to make sure, just as a disclaimer too, this is something we're all very familiar with because we live with this periodically throughout the year. I'm not here to solve a problem. I'm not here to give you answers to what we can do about the smell. It's more just sort of an exploration into some science and to some aspects of ecology that most of us don't think about very often. So with that, I'm going to share my screen. And if you have questions, please put them in the chat, and we should have time. I don't expect to talk for much more than half an hour, and we should have time, to answer a question in a little bit. So I'm hoping you can see this. Colleen, can you confirm that we've got the screen sharing going on here?
You are all set.
Okay, great, thanks. I can't see what you're seeing. So this is called seaweed microecology, or what I did my summer vacation. This was actually a research project that I underwent last summer, but it started somewhat before that. I think, oops, I'm just going to show you just a shout out to my school. This is where I work at Revere High School. I'm in my 11th year there, and I'm inspired by my students there all the time, even during our remote learning periods. And I just want to just show you these young people. They were actually not involved in this research. I had some other high school students I was working with, but I got my inspiration for education and for science from these fine young people. So we all have seen this. This picture was taken last April, about a year ago. This is up in the corner by on Lynn Beach near the Rotary, and you can see this mats and mats of brown, ugly seaweed that have washed up on the beach here. Now, it doesn't always look like this. I think there's a lot of dependency on current and tide and wind. That depends on whether or not this will wash up. And you may have noticed that it'll be there one day and then it will be completely washed away another day, even a few days later. So it's a bit transient, but this is our topic. What is this? What is this material? I just want to also give you. I'm going to try to move this little tabs here. I can't seem to bring up another tab. Let me see what I can do. So this is where we're talking about, for the most part. This is from Google Maps. This is Nahant Bay, and here we have Red Rock Park. This little area here is called Deer Cove, and the seaweed that we're talking about tends to wash up along this stretch. As a matter of fact, on this particular day when this shot was taken, you can see it washed up on the beach here. It just happens to be a bit of luck that we were able to get this picture. But this is really where we see it. It will accumulate further up the beach, and we see it as far as Fisherman's Beach, occasionally, almost never at Phillips Beach because it's facing in the wrong direction. But I'm sure we all have seen this and are familiar with it. Here's a couple of other pictures. It's when it lays up in these mats, it's about 1 centimeter in depth, and can accumulate quite a bit. With the tide, it can wash up onto the structures along the beach and dry up there. The interesting part, though, is that this in itself is not causing the smell. There's no relationship between laying up on the beach like this and the smell. The smell is caused by decomposition after it's buried in the sand, and I'll talk about that in a little bit. This is our organism here. It's called Pilayella litoralis, and I can tell you this is the most difficult name for me to say. It just does not flow off the tongue, and if I were going to be naming organisms, I would name them things that people could actually say. For the longest time, I was saying Pilayella, but a scientist at Northeastern corrected me and said that it's actually pronounced Pilayella. I found that even more difficult to say. But this is it, and this is an individual plant. It is a brown algae, a filamentous brown algae. It's in the same sort of group of algae as the kelps and the rockweeds, but this is a filamentous algae, and this is what it looks like in the water. Now, so I got really interested in this because, we live near it, and I'm a biologist, and it's just something that I find interesting. I'm a naturally curious person, so I've done a little research around this, and I'm sure some of you have too, and may know a thing or two about this, and maybe more than I do, and I'd love to hear about it. But in the scientific literature, this is really all I could find. This was a paper published in 1982 in a journal called Phycologia. I actually had to get the research librarians at Salem State to help me find this. It's not really easily available. This paper is called The Life, History, and Morphology of Free-Living Pilayella litoralis in Nahant Bay. Now, you may be aware of this. Some people will say this is the only place in the world where this exists. In fact, Pilayellas are extant throughout the northern latitudes of the world. They can be found pretty much anywhere in the cold regions. What's unique about Pilayella here is its form. It's a free-living form. In the rest of the world, Pilayella is sessile, which means it's attached to rocks or the substrate, the sand, or whatever. But here it's free living, and it also, at certain times of the year, can really bloom into these massive tons and tons of biomass. So it's not unique in the world here, but it's unique in its morphology and its way it reproduces. Here it has practically no evidence of sexual reproduction. It's pretty much all vegetatively produced, and we have lots and lots of it. I'd be happy to share this link with you. It's a very interesting article, and really the only scientific article I could find on this, which at first I was a little surprised by that. I thought, well, something that just causes such a stink, you'd think somebody would be looking at this. But, you know, I'm talking to scientists, they're like, well, it's a local problem. And scientists tend to not really focus on local problem things. It doesn't have global application. And so they're not gonna really be that interested in it, which is where somebody like a teacher or me or an independent researcher comes in, because we have the luxury of being able to not be bound by those restrictions. So this is our organism here, and I want to just shift gears real quickly. Now, some of you might recognize this is called an electron micrograph, and I wanted to just shift a little bit to give you a sense of where I got this idea and where I was going with this. This is actually a picture of a community of fungi and bacteria in cheese rind. Now, I never really thought about cheese rinds before until I went to a talk at Harvard by this woman. This is Dr. Rachel Dutton. She's at UC San Diego. She published an article in Cell Journal in 2014 about cheese rinds. And it was just fascinating. I was very lucky to get a chance to hear her talk to a group of teachers when she was at Harvard, and she brought all this cheese with her. Some of it was really stinky, which I'm not a big fan of stinky cheeses, but there it is. It turns out that cheese rinds are very, very complex microbial communities, bacteria, fungi, and that's what gives cheese its characteristic smell and taste, is those organisms. They develop over time as the cheese ages. When cheese is made, it only is inoculated with a handful of different bacteria, but as it ages, it grows into these very complex communities, and I just think it's really, really fascinating how that works. And so this is, again, this idea of a microbial community and a micro-ecology. They're all interacting with each other in ways to produce the smell and taste of the cheese. And then another professor I got a chance to hear was Peter Turnbull when he was at Harvard. And Dr. Turnbull is now at UC San Francisco. Everybody's moving to California. But he is one of the leaders and one of the original thinkers around the gut microbiome, which I'm sure many of you have heard about, that, you know, your gut is just filled with hundreds and hundreds of different kinds of microbes that assist you in digestion and all sorts of aspects of your life. And he's made connections of microbiome to all sorts of health problems. And so here again, we have this idea of a microbial community and how it affects the things that it lives with. Again, it's an ecology. We think of ecologies as like big macro-ecologies of animals and plants that interact that we can see, and that's because we're at that scale. But I'm here to say that there's a whole micro-ecology going on that we don't see, of many, many, many different kinds of organisms that are all competing and fighting and have mutualistic relationships and all the things that you would see in an ecosystem that are happening in a drop of soil, or in a gut, or in a block of cheese. So what does it have to do with this? So I drive by this every day, many of you do, and I got to thinking, well, first of all, it really smells, and secondly, it's huge and there's a lot of it. And so I started thinking there must be a really interesting microbial community happening here. And I think that what really spurred that on, of course, was the whole idea of the smell. Where is the smell coming from? It's some sort of hydrogen sulfide compound, and that rotten egg smell, I think that's pretty well understood. It smells different from that, so there's probably some other gases in there. But I started thinking about this as a community, not just as some yuck that lines the beach. So I kind of started to put some ideas together about how we might look at that. So science always starts with and ends really with questions. It's all about questioning, and I think that when I mentor young people about becoming scientists, I talk to them about, well, one of the first characteristics really that you would need is a natural curiosity. You really want to ask questions about things. And so in ecology or an ecosystem, one of the things you might ask is, what's in the ecosystem? What's there? And then secondly, what are they doing there? And those are the two big questions that you want to ask in an ecosystem. Who's there and what are they doing? How do they interact? How do they use energy? All these questions. And then the other questions I had were, can high school students do real science to answer these questions? I mean, real lab science using modern laboratory techniques. And then can these students also get access and use some of the cutting edge tools that we have in biotechnology? Now, why biotechnology? Well, if you want to know what's in an ecosystem, we're really fortunate now to have access to some tools in genetics that really allow us to look at the DNA of individual organisms. We call it environmental DNA. Think about, you know, a CSI show or something like that where they're trying to identify a suspect and they'll use DNA, and every individual person, as we know, has unique DNA fingerprint that can be identified. Well, that's true for all organisms. And so we can identify organisms by their genetic fingerprint. And in that regard, there's a very powerful tool that's been developed in the last few years by a company called Illumina. There are others. I'm not here to just plug Illumina, but they were very helpful to me in this process. There's a very powerful tool for analyzing environmental samples of DNA to identify organisms within those samples. So I thought, well, if we could get access to that sort of technology, you know, we could really go some places with this. So I was very fortunate to reach out to Illumina and did get some support from them, and I'll talk about that in a minute. And then, so if we want to identify organisms in a sample, there's really a couple of different ways to go about this. We're looking at the DNA and we're looking at the sequence of DNA. What that means is the unique combinations of letters, the A's, C's, T's, and G's, that are in DNA that allow you to identify one organism over another. Now, the old-fashioned way of doing that, which is not really that old, considering, you know, how long some of us have been doing this, we use a technique called Sanger sequencing. Again, this is getting a little sciencey. This talk was originally intended for a science audience, but what we do with Sanger sequencing is we target a unique sequence of DNA within an organism, and we do them one at a time. So you have to isolate those organisms first. So you have to figure out a way to isolate the organisms within the community, and then take that organism, isolate it, and then sequence its DNA. You do that one organism at a time. This method is cheap, it's relatively easy to do, and it can be done in high school. I do it in my biotech class. The new way is what we call next-gen sequencing. You can do hundreds of samples all at once. So instead of having to isolate individual organisms within a sample, you can take a heterogeneous mix, just a glob of dirt, and you can run it through this procedure. And in that process, all at once, you identify thousands of organisms. This method is not cheap, and it's not easy, and it's not something that is really accessible at the high school level. The old way is, and again, we've done that, and I can do that in high school. The new way, not so much. But I wanted to test this out and see what we could do with a bunch of high school students. Okay. I know I'm going fast, but Colleen, you can slow me down if I need to slow down. So I kind of put all this together in my mind about, okay, we've got the beach, we've got these genetics tools, we've got the seaweed, we have some work in the literature on microbial communities. How can I put this all together and make a project out of this? Well, one of the great things about being a biology teacher in this area is that we have access. I mean, really, I can't imagine teaching anywhere else, because we have access to public and private institutions that are really, really cutting edge in terms of our understanding of the natural world from a genetics point of view. I mean, Boston and Cambridge lead the way in the world for this research. There are other places. San Diego is one. University of Washington and St. Louis is one. There are others, but Boston is really it, and it's really the site of most of the genetic research. So I'm very fortunate to have some contacts in these institutions. And one place in particular, this is the biology building at MIT. And so I approached, this is where I ended up working in the basement all last summer. It was kind of dark and dreary, but we did get outside as much as we could. And I'm also very fortunate, I'm just going to give a few shout outs to this woman. This is Mandana Sasimfar. She is an outreach coordinator in the biology department there at MIT, and I've known her for many years. And she does great work with high school students and high school teachers. So I approached her and said, hey, Mandana, I've got this idea, you know, do you think you could help me out or figure out a way to make this happen? Well, the good news was it was actually serendipity, because Mandana also runs this program at MIT, which is a program for high school students from Boston and Everett and a couple of other, Chelsea maybe, a few other cities nearby,
that brings high school students in to do some microbiology and molecular biology research in the summer. They do that one summer, and then in their second summer, they get placed in a lab. And usually it's in a, like a research lab or a pharmaceutical lab or something like that. Well, she had some students that weren't placed yet. And she said, well, I can give you these students and maybe you guys can work together. And she also was able to get me some funding, which was super important. And she also, MIT isn't just going to let some random high school teacher come in there and start playing around in their lab. So she assigned this professor, Dr. Otto Cordero from the environmental engineering department, to be my sort of supervisor in this. He's the principal investigator. But Otto was great and very willing to let me work, and was a great mentor to the kids as well. So with that, I met these three students. Now this is Jessica. At the time, she was a junior, a rising senior at Boston Latin Academy. This is David. He was a junior at Everett High School, and Amog was a senior at Acton Boxborough. So I had these three kids, and we worked together in the lab all summer, and you can see stacks and stacks of Petri dishes from the work that we were doing. These kids were tremendous. I'm still in touch with them, and very, very pleased to work with them, and they really came a long way, and we all did. So we set up a lab down in the basement there and started to go to work. Well, the first thing we did is we went and collect samples. So this is us out in the field. This is Dr. Cordero and me and the kids. It was a great day for this because you don't see any, now notice you don't see any seaweed on the beach here, but boy, did it smell that day. This was a day in July and it smelled to high heaven. And that's when, we came to understand that the smell happens when the seaweed mixes in with the sand, and then it starts to rot. It starts to decay in the sand. So you can have a very smelly day with no seaweed on the beach. And in fact, right behind Jessica here, you can't see it, but in the little tide pools that we're developing, you could see the hydrogen sulfide gas bubbling up out of the sand. I've never seen anything like it. I wished I had had a hydrogen sulfide sort of detector with me to confirm that that's what those bubbles were, but my nose is a pretty good detector of these chemicals, as probably yours is. And so we were very amazed to see that stuff just bubbling up there. Just kind of like, it's like fermentation, right? If you ever made, well, if you make bread and you see the yeast rising, bubbling, or make beer, you see this bubbling happening. In this case, not carbon dioxide, it's hydrogen sulfide. And so we were fortunate to be able to see that. We didn't just look at Pilayella. We wanted to take a look at some other seaweeds. This is fucus. This is the rockweed here on the left. And this is ulva, the sea lettuce on the right. We also took samples from there as well. Okay, so what we needed to do though is figure out, we want to collect some microbes off of this algae, get some microbes out of this algae. And but first we were going to kind of go this old method of isolating organisms. So the first thing we had to do is devise a method for getting the bacteria off of the algae. Then you have to determine appropriate growth medium. How is this going to grow? Now, if you're familiar with a petri, I don't have any with me, but if you're familiar with a petri dish, it's just a plastic or a glass dish, and in the bottom it's got some growth medium in there, and you can grow stuff on it. And it depends, what grows on it depends on what you feed it. So usually in a lab, if we want to grow some E. coli or something like that, we use nutrient broth, which is basically a beef broth, and it will grow bacteria very nicely. But these are marine bacteria, and they need a special growth medium. So Dr. Cordero was able to get a hold of some algae, some marine growth medium for bacteria. And then I'll show you in a second, we streaked some plates under different conditions. We did them under cold conditions, warm conditions, room temperature, and we were able to isolate bacterial colonies and then isolate their DNA. And then once we isolated their DNA, we were able to go through this sequencing process to determine what bacteria we had in the dishes. So this might make more sense if I show this picture. This is 2 plates that we did. This is from Pilayella, and in this case, this was grown at 30 degrees temperature. This is a warm room sort of setting. We've also had cold settings. And each one of these little dots here represents a microbial or a bacterial, that's a bacterial colony. It's one type of bacteria. The technique is to streak it properly so that you end up with little colonies here. And then you pick those colonies off, extract their DNA, and send them off for sequencing. This one was from fucus. So we did that. Did a lot of this. We spent a lot of time this summer streaking and plating, and running what's called PCR, which you may have heard of in the context of COVID-19. It's a method for amplifying DNA and then sequencing it. So in the process of doing that, we came up with 17 different bacteria, and here are their names. So these probably don't mean a whole lot to any of you. They certainly didn't to me until I learned a little bit about them. I can't really tell you anything special about any of these, except the vibrios down here are very common in seawater. They're kind of the E. coli of seawater. Vibrio alginalticus, these are seaweed associated bacteria. So there is a lot known about some of these. Some of them have very brilliant colors. Some of them are cold temperature bacteria that only live in cold temperatures. So we grew some. We had some of the plates in the refrigerator, basically. And so, like the Cycromonas arctica, that is a cold-loving bacteria that will only grow under those cold conditions. And so we do see that in the cold water, even in the summertime in Nahant Bay. So this is what we came up with through this process. But I'm thinking there's got to be more to it than this, right? So the key point with something like this is you're only going to get what grows on the plate. So if the bacteria will eat what's on the plate, it'll grow, and you can analyze that. But there's tons of bacteria that are not going to grow in those plates because they don't have the right conditions. They don't have the right pH, the right temperature, the right salinity, whatever it is, the right food. They're not going to grow there. So that's where this idea of next-gen sequencing came in. So basically, for me, this was not enough. We only got what was growing on the plate, even though it was interesting. There was got to be a lot more out there. So this is where we get into working with Illumina. So Illumina makes sequencing machines. They do DNA sequencing. The picture on the right is from the Broad Institute in Cambridge. These are called HiSeqs. This row of machines here represents some of probably the greatest sequencing power in the world in that room. The high throughput, they can sequence a lot of DNA in a small amount of time. These machines are very expensive, millions of dollars. This room that is on Charles Street in Cambridge. It used to be a beer warehouse, and now it's part of the Broad Institute. And this actual location was where the Human Genome Project was done. Now, these machines weren't there at that time, but they are now. Now, Illumina also makes this little machine here that's called the iSeq, and that's a $10,000 machine sitting on a desk at the Whitehead Institute that they let me use. Now, it's not as quick, obviously, but it's certainly much easier to use. Illumina was very generous. They said, look, if you can find somebody who's got a machine that they'll let you use, we'll give you all the supplies and materials that you need, and we'll even send out a technician and he can help you do the sample. So they were great. I mean, really hats off to them for helping us out with this. The process of prepping these samples is difficult. It requires a lot of prepping. I had to use the services of the labs at MIT to do this, help us do this. It's expensive. Not something that schools generally are going to have access to. So that was good to know, right? So probably not something that I'll be arguing for grant-wise at school. But the results you get are truly, truly amazing. And I just want to share that with you. I think I have this up already. This represents the top 250 bacteria that we identified in our sample. There were over 700. The reads represents the relative frequency. And I want to point out this one, Paracoccus sulfur oxidans. It's the second most abundant one in our sample, and it really caught my eye because of its name, sulfur oxidans. It's a sulfur oxidizing bacteria. It produces hydrogen sulfide gas. I'm like, wow, look at that. We didn't even see that in our original study, but yet it's one of the most abundant bacteria in the sample. And in terms of next steps, I would really want to investigate this and see what's happening here and if this is the cause of our smell. Now, doing a little bit of research on this, so this particular bacteria is very interesting. It was first isolated in sewage sludge. And I guess that's not too surprising since sewage sludge often has a hydrogen sulfide smell. But what's it doing in Nahant Bay? Well, I think it's pretty clear. And again, this is a hypothesis, not a conclusion. We certainly have been surrounded by sewage treatment plants. Boston, the Lynn plant, Swampscott had a plant. And if I had to guess, and if I were going to take this into a new hypothesis, and that's again this whole idea of asking questions in science, I would ask, where did this come from? And and is it the cause of the smell? And then finally, is there anything we could do about it? If I had to guess, I would say that this bacteria has been with us a long time. The smell has been identified since about 1900. It was in the press mentioned, not before that. That doesn't mean it didn't exist before that. A lot of theories about, oh, they built the causeway and it caused this to happen, and we have no evidence of that, and really there's no way to know that. But my guess is that this bacteria has established itself in the Pilayella community and is there to stay. So I would, even with the cleanup of Boston Harbor and a lot better sewage treatment that we have now, I think we've got a, possibly even a really, what you might even think of as an invasive species that has established itself. Now, again, I'm totally speculating here. Don't take this as the truth, but I think that's where I would go with this, is try to figure out how to answer those questions. So that's really where we went with this, and I was just so thrilled. This was actually our very last week that we were able to work that we came up with this, and I was like jumping up and down to find this bacteria. I just thought it was a real find. So just a couple other things. I took the kids all over the place and had lots of collaborators. So we didn't just spend all our time in the lab. We had a lot of fun and had lots of guest speakers and a lot of support from lots of different people. Results, yes, there's a rich microbial community living within the marine algae. And yes, high school students can get involved in cutting edge biotech and biology research using the latest technology. This can absolutely happen. Again, if I were going to take this research further, I would want to look at some of the bacteria that are in these samples and try to identify what kind of interactions are happening there. Again, I'm not trying to solve a problem. Yes, the smell is obnoxious. Does it really affect me in my daily life? Not really so much. I mean, that's just me. So the next steps, I'd use the data to answer some more interesting questions. I would really want to work with industry and institutions to bring these technologies to school, up the game a little bit here. I think we could up the game some and expand the reach to more students and get more kids involved in these things, because these are real research questions that can be answered and students can get involved in that. I had a ton of collaborators. Very fortunate to have gotten to know a lot of people in this business, and people from Northeastern and Harvard and Salem State and really everywhere were willing to help out, and I can't thank them enough. And lastly, these three knuckleheads who, that's their last day selfie, who were just tremendous, and I really love them, and they were so much fun to work with. So that's my story, and I'll stop screen sharing.
Awesome, thank you so much. So we did have one request for the paper that you had shown earlier. So we'll figure out some way to either distribute that via email. I suspect we'll violate copyright if we post it anywhere, but we can probably distribute it via email.
From 1982, but
in some way, shape, or form. You do have one question from Margaret, and she asks, could Lynn and Swampscott collect the algae daily, take it away, and burn it?
The DCR has the control over the beach, and they do, in the summertime when it builds up, they come along with a front end loader and they pile it into piles, and then they take it to a landfill in New Hampshire. I talked to the DCR about this. They wouldn't give me any more specifics than that. They wouldn't tell me who the vendor is or who the landfill is, other than that's what they're doing with it. So that they are doing that. It is being taken away.
Awesome. If there's other questions, you can add them to the chat, or if you would like to unmute. It seems like we have a pretty docile crowd.
Yeah, I'm sorry, I hope I didn't put you all to sleep. But.
Oh. I don't know.
Yeah,
Sammy and Jack, are
Yeah.
you guys trying to ask a question? Go for it.
Yeah, so I have to unmute, right?
You are unmuted. We can hear you.
Thanks, Don. This has been really helpful. Does this occur anywhere else? When we first learned of the research way back when MIT was doing it with the big trailer on the beach and everything, we understood that it wasn't something that occurred anywhere else. Is that still the thought?
It doesn't to my knowledge and what I've read, it doesn't lay up on these big mats and create this big biomass really anywhere else. And mostly it's because it's this free living form that can live without being attached to anything. So it just floats around in the bay, and it reproduces just vegetatively and grows and grows and grows under, and it can grow, it grows all year round. It's not like just in the summer. And then under the right conditions with tide and wind, it'll just wash up on the beach. So there's really, the organism does exist throughout the world, but in this form, it's unique to Nahant Bay.
Thanks.
Yep.
Tony.
Go ahead. Go ahead, Tony.
Hey, hi, thanks, Don. That was great. You know, there was and probably will be a combined sewer overflow in Lynn, and there was also a problem with a storm drain in Swampscott that had sewage leaking into it that the town is spending a lot of money fixing. And both of those have probably been happening for a long, long time. And that's really close to the problem. Could this be a possible source of the, the sewage that's adding to the bacteria?
It could be, but I think the biggest concern that people have with the Stacy Brook and the combined sewer overflow is E. coli, because that affects human health. These other bacteria don't affect human health. So the only one that the EPA or anybody is caring or looking at is E. coli coming out of Stacy Brook. The interesting thing was we did not find any E. coli in our samples. Any. There was not a drop. So again, we took our samples from further down the beach towards Deer Cove. If you took them up by Stacy Brook, you might see some E. coli, but we didn't see any. So yes, those sewage sources could be a source of that. I was out at Deer Island and actually talking to a microbiologist there, and I'm like, "Do you guys ever look at what communities are in your digesters, what's in the digesters?" And they're like, "No, they don't research that at all." And at least the guy I talked to, he was a microbiologist, and he's like, "No, they didn't care." So, but yeah, that particular sulfur oxidizing bacteria could very well be in the combined sewer overflow.
Mm-hmm. And another question I had was, I don't know if this is happening, but it seems to be moving closer to Swampscott too. Is that possible?
I think it just over the years, like the tides, it ebbs and flows and just depends on the prevailing currents and the prevailing winds and how much biomass is generated through the course of the season. I wouldn't say that I'm not aware of a trend. I'd be surprised if there was any trend of it moving from one way or the other. It may grow in some areas and then move over to other areas. It's really going to depend a lot on a lot of different factors.
I have a question, Don. When the material flows up onto the beach, is it dead then, or when the next tide is it alive and it just goes back out alive?
It's a great question, Jim. My guess, and again, I haven't found any research in this and we didn't look at this. My guess is there's a fair amount of living tissue, living material in the stuff that's on the beach and a fair amount of dead material. It's probably a mixture of both. I don't think it's not washing up on the beach because it's dead. It's washing up on the beach because it got blown there. And if it sits up on the beach for too long and it gets baked in the sun, it'll die. But it's not coming up on the beach because it's dead.
Thank you.
Yeah.
Awesome. Other questions?
Is Martha on here? Does she have a question?
No. Tony wants to ask another one. I'm going to unmute her. I like having the power to mute Tony.
I know you do. Does anything feed on it?
I would imagine that there's a fair amount of feeding that's going on amongst the different arthropods that are living in there. So arthropods, like sometimes you'll see these little, shoot, I'm losing the name of them. They're like little tiny shrimpy guys. Colleen, what am I talking about here? You know what I mean? If you ever pulled a rope off a mooring, you'll see them spilling off of it. These little guys, and they're definitely feeding on it. I'm guessing the little critters that are in the water are eating it. Sure. I don't think it's like a major consumer of it, but it's going to be the marine arthropods that are mostly eating that.
So you have a question from Martha in the chat, and she's asking how it reproduces.
So it doesn't in Nahant Bay anyway. It doesn't have any sexual reproduction. So it's not producing any fruiting bodies or anything like that. In other parts of the world, it does. In Nahant Bay, it grows vegetatively. It just does asexual reproduction. Just grows and then it buds itself off. So it'll grow to a certain size, and then it'll just break off, and then that one will grow and then it'll grow. And they get to be maybe 2 or 3 centimeters. If you put them in water, they form like a ball. And they get to be maybe 2 or 3 centimeters in diameter, not very big, but they just kind of bud off and grow from there. They just spread like weeds.
Okay, maybe one final question if there's anybody out there. No? Then I think we will say thank you to Don for a wonderful evening. We can do some virtual clapping or some snapping. I see those with video on applauding you, Don. Hopefully you can see them as well.
Well, I appreciate that. I just wanted to say, I just find it a really interesting topic. I mean, it's something that we live by and we were very close to. And I think that if you look around, there's all sorts of questions that you can ask about the world that's around you. And this one might not be the sexiest one, but it's out there and it's kind of interesting. At least it was to me. So there's all sorts of stuff you can ask.
That's very cool. We really appreciate you taking the time to share the story with us. We thank everybody for tuning in tonight, and you can find out more about the Swampscott Conservancy on our website, which is just our name, so swampscottconservancy.org. You can also feel free to email us at swampscottconservancy@gmail.com if you are not already on our email list. And you can also follow us on Facebook. And with that, I think we will say goodnight. So now you can hopefully go answer that question. Oh my gosh, what is that smell? The next time you head to the beach. And hopefully that's the only debate that we'll have this summer when we go to the beach. How about that for a topic?
All right, thank you, everyone.
Good night, everybody. Thank you.